In this video, we'll be continuing with our British Biology Olympiad preparation, specifically looking at biological molecules and carbohydrates as our main focus for today. I do have part 1, part 2, and part 3 of the video series on carbohydrates, so I highly recommend that you check out those videos prior to watching this one because it contains a lot of prerequisite knowledge that I think is really important for comprehending the content dealt with in this video. Right, so first of all, we're going to look at the disaccharide maltose. Now, maltose is a disaccharide that's composed of 2 glucose units. More precisely, it's composed of 2 alpha D-glucose units when it forms a 1,4 glycosidic bond. Now, this is one of the 2 main types of glycosidic bonds that we will be dealing with, the 2 most common, 1,4 glycosidic bond and 1,6 glycosidic bond. But to emphasize, it's dealt with 1,4 glycosidic linkages. This basically means that a bond is formed between carbon 1 of the first glucose molecule and carbon 4 of the second glucose molecule. And we saw how this is a condensation reaction because we get the removal of water, specifically involving the removal of the hydroxyl group of the first glucose and the hydrogen group, or the hydrogen of the OH group of the second glucose. So we get removal of 1 water, and we're going to get this bond specifically concerning an oxygen within the bridge, oxygen atom within the bridge. Now, because a glycosidic bond kind of involves the anomeric carbon of only 1 glucose unit, the second glucose, this one, is going to still retain a free anomeric carbon. The anomeric carbon refers to carbon 1 or C1 right here. And as a result, maltose is going to be classified as a reducing sugar. I think this is very important because reducing sugar, we've talked about this concept a long time ago, I think, also in IGCSE, but we haven't really went into a lot of depth about what it is. Reducing sugars are basically sugars that have free ketone or aldehyde groups, and basically these can kind of donate electrons is what it is. So if I had to cite a couple of examples regarding reducing sugars, it would be glucose. We would have fructose as well, I think, and maltose as well. And specifically talking about the Benedict's reagents test, where we have to add a couple of drops of Benedict's reagent and then kind of observe color changes. This reducing sugar is what causes the color change from, I think it was blue to brick red. So it's the reducing sugars that are responsible for this sort of chemical change in the Benedict's reagent, just as a side note. So if we look at the molecular formula again for maltose, we can kind of see how one molecule of water has been removed in the chemical equation. It's C12H22 and O11. When we double the molecular formula of glucose, we can get C12H24 and O12. We see that the difference here is regarding an H2O, so we can see that a water molecule has been removed in the process of forming this condensation, going through the condensation reaction to form the 1,4 glycosidic bond. So we can see that chemically as well. Right. Second disaccharide that we're going to talk about is sucrose, and it's formed when 1 alpha glucose molecule and 1 fructose molecule kind of bonds together. And the 2 monosaccharides are joined by a 1,2 glycosidic bond. This isn't necessarily, I guess, the most common, but a 1,2 glycosidic bond specifically concerning the first carbon of a glucose, the alpha glucose, and carbon 2 of fructose. They join together in this manner in order to form a 1,2 glycosidic linkage. And in sucrose, both of the anomeric carbons are involved in the glycosidic bond. This means that there's no free anomeric carbon, and as a result, sucrose is not a reducing sugar. It's a non-reducing sugar. And like maltose, sucrose has the chemical formula of C12 and H22 and O11. But still, like, despite having the same molecular formula, I guess the nature or the chemical properties of it differ significantly because of the nature of the bonds and the monosaccharides that are involved. Right. Third disaccharide that we're going to look at is lactose, and it's formed by 1 glucose molecule and 1 galactose molecule. And these monosaccharides are joined by a 1,4 glycosidic bond that's formed between carbon 1 of the galactose molecule and carbon 4 of the glucose molecule. And because only one of the anomeric carbons are involved in the bond, lactose is again has a free anomeric carbon, and therefore it is also a reducing sugar. And lactose is most commonly found in milk and dairy products, and the molecular formula is again the same, C12H22 and O11. You've probably heard about lactose in the context of having like lactose intolerance, and that is the same lactose that we're talking about, the one that's in milk. Right. Next, we're going to look at polysaccharides. And polysaccharides are basically large carbohydrate polymers that are composed of many monosaccharides that are joined together using glycosidic bonds. The key polysaccharides that are studied here, specifically in the context of IB biology and also, I guess, in the BBO, is for starch, which functions as a carbohydrate storage in plants, and we have glycogen, which is basically carbohydrate storage in animals, and cellulose, which essentially is the sort of structural component for plant cell walls. And although in all of the three polymers, they're all three polymers of glucose, the properties do differ because of the type of glucose that is used and the type of glycosidic bond that is present in each of these structures and the degree of branching for each polymer.